cancer or metastasis, it is possible to remove a large part of the liver, including the tumors, and then the remainder of the liver can regenerate. As you can see, the liver is located very close to the stomach. The bile duct is released, the content of the bile via the bile duct is released in the small intestine, the duodenum, and then it helps to digest especially fatty components of the diet. At a more microscopical level, you can see that here is the entrance of blood from the intestine, the portal vein branch. So there are a kind of units that repeat itself throughout the liver. This is one unit, and these units, these anatomical units, are repeated throughout in the liver and make up the liver. The blood enters via the portal vein, the blood from the intestine containing all the nutrients. Oxygenated blood enters via the hepatic artery, and then the blood flows via this system of specialized veins, I would say, blood channels called sinusoids. It collects again in the central vein, and then it is transported further via the portal, via the vena cava. Now, what is very important is to introduce to you the different cell types in the liver, because I will come back to that quite extensively. The most abundant cell type in the liver are the hepatocytes, that are these square cuboidal type of cells, and these are the cells that make the liver the liver. They contain the liver specific functions. The liver functions, I will come to that later, but all the enzymes and the machinery to metabolize nutrients into useful components for the human body are performed in the hepatocytes. What you can also see in the sinusoids is that normally blood vessels are lined by vascular endothelial cells or arterial endothelial cells. In the liver, this is a little bit different. There are endothelial cells lining the hepatic sinusoids, but they are very different from the endothelial cells that line arteries and veins. They are more porous. And the reason is that because there are pores in these cells, they can exchange nutrients from the blood and toxic waste products from the hepatocytes more easily. In this part of the figure, you can see where these endothelial cells are located. So this is the sinusoid. This is the same as this part, and they are lined by these. Yeah. And they are lined by sinusoidal endothelial cells. In the vascular system, the endothelial cells form a continuous lining, but here you can see that there is space between the endothelial cells, facilitating exchange of nutrients to the hepatocytes and from waste products to the blood. And in addition, these cells also contain pores that allow exchange of these components. What you can also see inside the sinusoidal lumen, so in this area, are so-called Kupffer cells. Kupffer cells are macrophages specific for the liver, and they are the first defense against invading pathogens and toxic components that we ingest via food or that is present in the blood circulation. So the Kupffer cell is able to let's say unharm viruses, bacteria, fungi, and other pathogenic microorganisms. So the Kupffer cells are macrophages specific for the liver. Between the sinusoids and the hepatocytes, so here in the figure, there is a space. You can see a space behind the endothelial lining. And in this space, which is called the space of Disse, we see stellate cells. Stellate cells have two phases. You could compare them to fibroblasts. In normal conditions, in healthy livers, the stellate cells are very important in the metabolism of lipids. So they store lipids, they store vitamin A, and so they are very important in the metabolism of lipids. In diseased conditions, these cells show their other phase. They become fibroblasts, they start to proliferate, they start to produce scar tissue, extracellular matrix, which is accumulated in this space. So it clogs this space completely, and therefore the exchange of nutrients and toxic waste products is interrupted, because we will see that in a moment, because this space is completely filled with extracellular matrix in chronic liver diseases. What you can also see here are the bile ducts. So the hepatocytes produce bile, and the bile that is produced in the hepatocytes is transported into these bile canaliculi in green here, which is basically here. And then these bile canaliculi, they drain into these bile ducts, and all these bile ducts in the liver eventually come together in the bile duct. So the formation of bile is on the other side of the hepatocytes, that is faced towards the bloodstream and the sinusoidal lumen. Now, what are the functions of the liver? The liver has many functions, especially in, let me see if I can get rid of this. First, one of the most important functions of the liver is the detoxification. So it is the organ in the body that is exposed the first to toxic components. For example, if we eat food which contains harmful components, then via the portal vein, the liver is the first organ that sees these toxic compounds. Also, when we use medications, drugs, alcohol, it's taken up in the blood, and it is metabolized and taken up and metabolized in the liver. So, for example, if you use very common painkillers like paracetamol or acetaminophen or naproxen or ibuprofen, all these drugs are metabolized in the liver because they are considered as strange components. And the waste products, the metabolites of these drugs, are excreted into the bile or transported to the urinary system. The liver is not the only, but the most important organ that metabolizes alcohol. It has several enzyme systems that can metabolize alcohol, but the problem is that the metabolites of alcohol cause liver damage. So many of these detoxification mechanisms work very well when you use moderate amounts of drugs and alcohol, but if you use excessive amounts of drugs, for example, acetaminophen overdoses or long-term chronic alcohol consumption, the metabolites of these drugs and alcohol cause liver damage. In addition, pathogens that enter the bloodstream, they are via the portal vein also transported to the liver, and there the Kupffer cells, I already mentioned these cells, they recognize pathogens like viruses and bacteria, and they kill these viruses and bacteria. Unfortunately, there are also viruses that can infect specifically liver cells, and you all know hepatitis B, hepatitis C. These are viruses that can bypass the defense mechanisms of the Kupffer cells and the immune cells. They can infect the hepatocytes, take over the machinery of hepatocytes to to produce viruses, and this causes also liver damage. So hepatitis B and hepatitis C viruses are viruses that can infect the liver cells themselves. Another important function of the liver is metabolism. So all the proteins, the lipids, and the carbohydrates that are ingested in the diet, they are broken down in the digestive tract into all kinds of intermediate products, amino acids, free fatty acids, sugars, and these are taken up. This is done in the liver. So this metabolism of proteins and lipids and carbohydrates takes place in the liver, and then these components are used for the body to produce new proteins. If there is an excess of sugar, it is stored as glycogen in hepatocytes, and lipids are also converted into other lipid species that are useful for the body, or the excess fat is the excess lipids is stored in adipose tissue. An important component that is produced from lipid species and bile acids is cholesterol. Cholesterol, we all think cholesterol is bad because high cholesterol levels in your blood is dangerous for atherosclerosis. But cholesterol is also an important building block of cells. The cell membrane contains a lot of cholesterol. Cholesterol is the most important component in cell membrane. So we do need cholesterol. But our diet is sometimes excessive in lipids and cholesterol, and therefore we also have sometimes high levels of cholesterol in our blood. But it's not a bad component per se. And cholesterol is built from bile acids and lipid species and other components. Protein synthesis. As I mentioned, the liver is the site where proteins are broken down in amino acids and used for synthesis of novel proteins. And many of the proteins that are synthesized by the liver end up as plasma proteins in the blood. So almost all proteins involved in blood coagulation and blood dissolution, for example, fibrinogen, alpha-1 antitrypsin, complement factors, prothrombin, all these proteins are produced in the liver. Another very important protein that is produced by the liver is albumin. Albumin is the most abundant protein in blood, and it maintains the osmotic value of the blood. If you have low albumin levels, it is usually a sign of liver damage, and also very dangerous because your oncotic pressure, your osmotic value of the blood is compromised. Lipoproteins, which are involved in transport and metabolism of lipids and lipoproteins, are also produced by the liver. So it's one of the most important sources of novel protein synthesis in the body, the liver. The liver also produces bile, and it uses cholesterol as a building block of bile. But many of the bile acids contain a cholesterol skeleton, but also they have several amino acid groups or even sugar groups attached to it to make it more soluble. Excess carbohydrates are converted in sugars, and the sugars are converted into glycogen, which is, let's say, a kind of starch, which is accumulated in the hepatocytes. And can be used as a reservoir if there is insufficient energy, insufficient carbohydrates. And of course, the carbohydrates themselves, the sugars, are used for energy production in the mitochondria. Liver is also very important in the storage of micronutrients, both minerals like copper and zinc and magnesium and iron, but also vitamins. This also means that if something goes wrong in the metabolism of, for example, copper or iron, it can give rise to very specific liver diseases. There are genetic diseases of the liver which are related to let's say, incorrect storage of copper and iron, and this can lead to very serious liver diseases necessitating liver transplantation. The liver, although we do not think of the liver as an immune system or an immune organ, it does contain a lot of lymphocytes, natural killer cells. So it's an important component of the immune system as well. It is involved in the recognition of pathogen together with macrophages, but it also contains a lot of immune cells. So you can understand that the liver is crucial to many central processes in the human body, and therefore liver diseases usually affect many other organs. So if your liver fails, you will have problems with, for example, blood coagulation, blood coagulation and fibrinolysis of blood. You will have problems in the metabolism of toxic compounds and alcohol. You will have problems in production of certain proteins or lipids, so a lot of metabolic disorders. So therefore, liver diseases can be quite serious diseases. Now, which liver diseases do we know? I hope. Yeah. I already mentioned that alcohol is a very important source for liver diseases. Fortunately, drinking moderately once a week or so is okay, but if you drink for a very long time, excessive amounts of alcohol, your liver get damaged. The reason is that alcohol is metabolized by the hepatocytes into metabolites that cause damage to the hepatocytes. So they generate toxic metabolites. The same is true for exposure to drugs and toxins. So drugs that are very well known that are harmful for the liver when used in excessive amounts are paracetamol, so acetaminophen, diclofenac, ibuprofen, and many other drugs as well when used for a long term or in high doses. There are a lot of genetic disorders that cause liver diseases. I already mentioned a few. There are genetic disorders in which the metabolism of copper and iron is disturbed. This leads to the accumulation of copper or iron in the hepatocytes, and this causes liver cell damage to the hepatocytes, and this causes then liver injury. I also mentioned the viral hepatitis B, hepatitis C. Nowadays, there are vaccines available for hepatitis B, and there are drugs available for hepatitis C. So it is expected that the incidence of viral hepatitis will decrease, but this also depends on, for example, the availability of drugs for hepatitis C, but also the availability of vaccination programs for hepatitis B. One of the most important causes of liver diseases in the world nowadays is obesity and fatty liver. We all know that our lifestyle in the whole world changed dramatically over the past 50 years. We exercise less, we perform less physical activity, we do less exercises, and we eat more food, more unhealthy food. We eat more McDonald's burgers and Coca-Cola than healthy fruit and juices. So we are exposed to an overdoses of carbohydrates and fats, and it's not used as much as it, because we don't perform that much of physical activity. So the excess of carbohydrates especially, but also lipids, translates into a liver condition which is called fatty liver. And fatty liver is the accumulation of fat in the hepatocytes. Now you can imagine that if you accumulate fat in hepatocytes, it will take up a lot of space inside the cells. And this will cause a lot of stress to all the organelles inside the hepatocytes, leading to death of hepatocytes and eventually chronic liver injury. Nowadays, about 30% of the world population has some form of obesity, some form of overweight. And it's expected that this will rise more in the future. And also the cases of extreme obesity, so morbid obesity, is also likely to increase very much. All these people with obesity and fatty liver can develop liver injury and eventually liver failure. So you can imagine that if 30% of the population has a fatty liver, that we will have in the future an enormous amount of patients with serious liver injury and liver failure, and they will all need liver transplantation. So this is going to be one of the future health problems, the shortage of donor livers. The only cure for fatty liver is to lose weight. And we have now, for example, drugs like Ozempic, which at least in the short term seems to work to lower weight. There is one drug approved for fatty liver, which is still not very widespread, but even with the availability of these drugs, we expect that the number of patients with a fatty liver needing liver transplantation will rise. A complication of obesity and an excess intake of sugars is diabetes. So diabetes, obesity and fatty liver, and an unhealthy diet are all related. The final group of liver diseases that I want to mention are autoimmune liver diseases. So we have autoimmune diseases in many organs. Basically, in all organs, you can have autoimmune diseases. For example, chronic inflammation of the intestine, the Crohn's disease, ulcerative colitis, they are thought to be autoimmune diseases, or at least there is an autoimmune component in it. Rheumatoid arthritis is an autoimmune disease, and there are also autoimmune diseases in which hepatocytes are attacked by the immune system. And these diseases are quite serious, not very frequent. It's not a major cause of liver diseases, but there are many examples of autoimmune diseases in which the liver is attacked. Usually the drugs that are used for autoimmune diseases are immune suppressive drugs like methotrexate or azathioprine. So there are quite a lot of liver diseases, but the fatty liver is going to be the most concern. Now, if we talk about liver diseases, there are several stages. Like in any disease, there are several stages of disease. Here you see the healthy liver, and it has a brown color because of the iron storage function of the liver. So the brown color of the liver is mainly iron. At early stages of most liver diseases, there is a stage in which fat is accumulating in the hepatocyte. The liver still functions, but the accumulation of fat or the metabolism of alcohol or the metabolism of overdoses drugs lead to a kind of inflammation. And if the inflammation is chronic, we will get the more serious stage of fibrosis. Fibrosis means that the liver is accumulating scar tissue. So I already mentioned in the beginning that an important cell type in the liver are the stellate cells. They act like fibroblasts. And in conditions of chronic liver injury, these fibroblasts start to produce scar tissue, extracellular matrix. And this is accumulating in the liver, and it's accumulating to such an extent that it is basically pushing away, killing functional liver mass and replacing it by scar tissue. So this is a very serious condition in which your liver loses function and in which the liver is replaced by scar tissue. So the liver also becomes very stiff. If the fibrosis is very advanced, we speak of cirrhosis. In cirrhosis, this process is not longer reversible. Here, if we take away the cause of the injury, the liver can still recover. But at the stage of here, advanced fibrosis and cirrhosis, there is no way back. There is only the possibility for transplantation. Moreover, in the cirrhotic stage, the remaining hepatocytes that are left are trying to compensate for the loss of hepatocytes, so they start proliferating continuously. And you know that if you have continuously dividing cells, you have a higher chance of cancer. So remember that when I talked about fatty liver as being the most prevalent liver disease of the future, 30% of the population has overweight, is obese. Many of these patients will develop a fatty liver, fibrosis, cirrhosis, and eventually liver cancer. So we will also face in the future an epidemic of liver cancer, hepatocellular carcinoma. So our lifestyle with regard to the liver will predict a lot of troubles. In addition to other complications of obesity like atherosclerosis, diabetes, and dementia. Now, I'd like to discuss in a little bit more detail what is going on when you have liver injury, because that is what we are studying at the University of Groningen, the mechanisms of liver injury, liver inflammation, and fibrogenesis. Here we have a scheme at the cellular level. So again, we have the healthy liver, normal liver. We have the injury, which can be virus, alcohol, bacterial products like endotoxins or lipopolysaccharides, excessive bile acids. If your bile acids are not removed from the liver but are blocked, for example, if you have gallstones or cholestatic liver diseases, then bile acids accumulate in the liver and can also be toxic. Accumulation of iron, of copper can lead to liver injury, excess free fatty acids and glucose as well. Now, what happens in all these cases of injury is that you get an inflammatory response in the liver. And an inflammatory response means that your inflammatory and immune cells become activated. So the Kupffer cells, the macrophages of the liver, become activated and they start releasing toxic components like reactive oxygen species and inflammatory cytokines that act on the neighboring cells, for example, the hepatocytes and the stellate cells. So the activated Kupffer cells in inflammation can damage hepatocytes, and at the same time, they can activate the stellate cells, which normally are responsible for the storage of lipids and vitamin A. But when they become activated, they start to proliferate and they start producing scar tissue or extracellular matrix. You can see that here. This is the resting stellate cell, quiescent stellate cells. You see the lipid droplets containing vitamin A, but in inflammation, it becomes activated, it loses its lipids, and it starts producing extracellular matrix composed of collagens, for example. Also, the Kupffer cells become activated. They release enzymes that inhibit the breakdown of extracellular matrix, so amplifying the deposition of extracellular matrix within the liver, leading to liver damage. And then we speak of fibrosis. Here you see it again. This is the normal liver, the main liver cell types. Chronic liver injury, all cell types undergo phenotypic alterations. The hepatocytes become damaged, so they lose their microvilli, which are projected towards the sinusoid, so they are inhibited in the uptake of nutrients from the blood, from the sinusoids. The stellate cells that are quiescent in the normal conditions become activated, so they proliferate. You can see that here. And they also deposit scar tissue within the sinusoids, within the space of this, excuse me. The Kupffer cells also become activated. They start producing reactive oxygen species, inflammatory cytokines, amplifying the effect on stellate cells and the damaging effects on the hepatocytes. And finally, the endothelial cells, where you see, let's say, spaces between the endothelial cells, these spaces become continuously. So these endothelial cells are starting to make a continuous lining, and that will block the exchange of nutrients to the hepatocytes and the removal of toxic metabolites from the hepatocyte to the sinusoid. So this loss of spaces between the endothelial cells and the accumulation of scar tissue in this space hampers the exchange of components between hepatocytes and the sinusoids. And that leads to a lot of complications not only in the liver, but in the body as a whole, because your whole metabolism is disturbed. I would like to focus a little bit more on steatotic liver diseases, or fatty liver, because it's the most prevalent liver disease nowadays, and its prevalence will only increase in the future. So what are risk factors for fatty liver? Apart from an unhealthy diet, we already talked about it, overnutrition with a poor quality diet, rich in sugars, carbohydrates, fructose, glucose, and saturated fats. That is a risk factor for developing fatty liver. Also, alcohol drinking is a risk factor, but there are also genetic factors. So certain genetic variants imply a higher risk for fatty liver than other variants of these genes. For example, the PNPLA3 gene, which is a gene involved in lipid metabolism, certain variants of that gene predispose to steatotic liver diseases. And then you can also have an increased risk of fatty liver if you have obesity, which is related to the unhealthy diet, diabetes, but also certain disturbances in lipoprotein metabolism. So hypertriglyceridemia and dyslipidemia can also be risk factors for steatotic liver diseases or fatty liver diseases. Again, we have different stages. So fatty liver diseases, we have about 30% of the general population which is in this stage, fatty liver. And of this stage, a part will continue to develop chronic inflammation. So the hepatitis stage, the inflammation stage of fatty liver, and this can develop, chronic inflammation can develop into fibrosis and cirrhosis, which is partly irreversible, especially cirrhosis is not reversible, and this can lead eventually to cancer. So again, if you consider that 30% of the world population is here, you can imagine how many people we will have with irreversible liver damage and cancer in the future. So it's really a serious disorder, and it's also an area in which we investigate a lot in our laboratory in Groningen. How does it look like a microscope? Well, this is a microscopical image of the normal liver. You can see here in pink, you see mostly the normal hepatocytes. The dark blue here is the nucleus. But what you see here, all these cells are healthy hepatocytes. Hepatocytes are approximately 60% of the cell population in the liver. The hepatocytes are the cells that make the liver the liver. And you can also see that the liver is of a normal size and a normal color. If you have fat in your liver, the fat is accumulating as lipid droplets in the hepatocytes. So what you see is that these lipid droplets are very big. They are bigger than a hepatocyte. So the hepatocyte that is accumulating these lipid droplets, they get bigger, they enlarge, and that induces a lot of stress on all the organelles in the hepatocyte. So you can easily appreciate that if you have this size of lipid droplets in your hepatocytes, that your hepatocytes are not doing very well, and in the end will die and will get damaged. This is the stage of fatty liver without inflammation. So this is what 30% of the world population has. This is, by the way, an advanced state of state. So I would not say 30% of the world population has this, but is on its way to this stage. Then if you get damage to your hepatocytes, activation of your immune cells and macrophages, your Kupffer cells, you get inflammation. And inflammation is the infiltration of inflammatory cells, macrophages from the blood that infiltrate the liver, and together with the Kupffer cells, the liver-specific macrophages, they produce a lot of cytokines and reactive oxygen species that damage the hepatocytes. This is the stage which we call steatohepatitis. Steato means fat, hepatitis means liver inflammation. This is the stage where you have inflammation as a result of the storage of fat in the liver. And if this stage is chronic, if you don't lose weight or lose your lipid droplets here, if this becomes chronic, then you develop fibrosis, cirrhosis, and in the end, liver cancer. Here you see a larger magnification. You can really see that these lipid droplets are huge. This is the nucleus of a hepatocyte, and these lipid droplets are really huge. And here you see the infiltrating macrophages causing the inflammation in the liver. Let me see, 42 minutes, yes. So I will now change a little bit to research that we are doing in our laboratory. You can consider this part as an introduction to liver and liver diseases. I will briefly demonstrate a little bit the research that we are doing in our laboratory. And we are mainly interested because if you have a liver disease, as I mentioned, all cell types in the liver change their behavior. You have phenotypic alterations in all liver cell types. And these cell types also have influence each other. So they talk with each other. There is communication between different cells in the liver. And in liver diseases, this communication is disrupted. Normally people talk with each other if everything is right, but in some conditions, in disease, pathological conditions, people can start screaming to each other, and that can inflict danger or intimidation or damage to other people. And the same is true in the liver in chronic inflammation. If these macrophages and immune cells in chronic inflammation become activated, they start screaming at stellate cells and hepatocytes, and you have a lot of conditions, a lot of miscommunication, and things go wrong in the liver. I already showed this slide in the previous part, but again, if we go from the normal situation here to a diseased situation here, we see that the hepatocytes, the functional liver cells, lose their function and lose their structure. We see that the Kupffer cells become activated and start releasing reactive oxygen species and cytokines, which impact on other cell types, usually in a damaging way for hepatocytes or in an activating way for stellate cells. So the activated Kupffer cells activate the stellate cells, and activated stellate cells produce scar tissue. So activation of stellate cells is not good. We want our stellate cells to be in the resting state where they store lipids and vitamin A, and we don't want them to become activated, proliferating, and producing scar matrix. Also, we see that the sinusoidal cells lose their structure, inhibiting exchange of nutrients and waste products, and also immune cells, NK cells, NKT cells become activated. In our lab, we are, I will skip this for the moment. In our lab, in our laboratory in Groningen, we are interested in how these cell types influence each other during the development of liver diseases. I already mentioned hepatocytes, Kupffer cells, the liver-specific macrophages, the sinusoidal endothelial cells, the endothelial cells that line the sinusoids, the small blood vessels of the blood canals in the liver, and stellate cells. In our laboratory in Groningen, we are able to isolate all these liver cell types from rat liver. So we can isolate the different cell types from the liver, and we can put them in culture. What you see here are all cultured cells. These are cells which are isolated from rat livers, separated from each other, and brought into culture. So these are cultured hepatocytes, cultured Kupffer cells, cultured stellate cells, and cultured sinusoidal endothelial cells. And these cells communicate with each other. And usually people look at two cell types for intercellular communication. For example, they investigate what the effect is of Kupffer cells on hepatocytes, or they investigate what the effect is of Kupffer cells on stellate cells. But in reality, all cells communicate with each other. The Kupffer cells do not only communicate with stellate cells or with hepatocytes, but also with the sinusoidal endothelial cells. And the hepatocytes communicate with stellate cells. And then another layer is that this communication is not one way. Here we see the arrow pointing towards the hepatocytes, but in reality, there are also arrows going to the other way. So you see that this intercellular communication becomes quite complex because this cell influences this cell, but this cell will also influence this cell type. And the final layer of complexity is that the communication changes when from a healthy state to a disease state. For example, during liver inflammation, Kupffer cells become activated. So the signals they produce in the inflammatory state, in the activated state, are different from the signals they produce in the healthy state. So we have a very complex interplay between the different liver cell types in the normal liver, but also in the diseased liver. So this is quite complex to study. How do they communicate, liver cells, between each other? Of course, you can think of factors that are produced by cells, secreted by cells, and then they go in the space between cell types in the liver. They travel by diffusion or whatever from one cell type to another. And that can be like single factors produced by cells, for example, cytokines or reactive oxygen species. In the last 10-20 years, we have discovered, well, the community that investigates liver diseases has discovered another way of communication, and that is communication via extracellular vesicles. Extracellular vesicles are small lipid-surrounded vesicles that are released from the cell membrane of cells, and they contain content of the cells that are releasing these vesicles. So the content of these cells can be a lot of different things. It can be proteins, it can be RNA, it can be enzymes, it can be hormones, it can be cytokines. So they are released from the cells. You can see that here by budding from the cell membrane, and they take with them part of the content of the cells. And these vesicles can interact with other cells. They travel to another cell, and then the membranes fuse, and the content of these vesicles, which is actually the content of this cell type, is released in the target cells. There are different types of extracellular vesicles. based on their size. And we focused mostly on exosomes. Exosomes are the smallest type of extracellular vesicles. There are also other types available, but I will focus mainly on exosomes because they contain a lot of the, let's say, bioactive compounds that are released from these cells and target on target cells. By the way, thinking of a biomedical engineering applications, people have also been using these exosomes to add specific drugs or compounds in these vesicles as a kind of therapeutic nano approach. So that is a very exciting field, but we don't do research on that. But maybe in one of the future lectures, this will become more discussed in more detail. Once again, extracellular vesicles, they are released from the source cell. They contain components that are taken from the source cells, and they travel to target cells that can be hepatocytes, stellate cells, Kupffer cells, sinusoidal endothelial cells. The content of these vesicles, as I mentioned, are proteins, enzymes, RNA, DNA, exosomes, but also hormones and cytokines. And when they reach the target cells, their membranes fuse and the content of these vesicles is released into the target cells. This way of communication has been very much studied in the last 10, 15 years. And we think that it is a very important way of communication also in the liver, in the context of liver diseases. What is very important to realize is that the content of these vesicles change in the course of disease. So in normal conditions, vesicles released from cells in normal conditions have a different content than when they are released from, let's say, cells in a disease state. So again, using the Kupffer cells, the macrophages as an example, the content of extracellular vesicles released from Kupffer cells in a normal liver is different from the content of the vesicles released from activated Kupffer cells. The same is true for stellate cells. The content of vesicles of resting quiescent stellate cells is different from the content of activated stellate cells. More general, extracellular vesicles are not, let's say, only limited to the liver. They are produced by all cell types and therefore by all organs, and they can be found in all, let's say, fluids of humans. So they can travel short distances between cell types in one organ, but they can also be released in the blood. So they can be used as biomarkers. So the next lecture, or one of the next lectures by Dr. Camila Trios, will be on the use of extracellular vesicles in blood as novel biomarkers. They can be found in urine, breast milk, saliva, cerebrospinal fluid, and they have been implicated to be involved in all these diseases, from neurodegenerative diseases to autoimmune diseases and steatosis and fibrosis. We can also isolate extracellular vesicles, for example, from blood, but also from culture media if you culture cells. So this is an example for the isolation of vesicles from blood. It is isolated by a series of centrifugations and ultracentrifugations. And in the end, we have a small amount of extracellular vesicles, which we can analyze by mass spectroscopy, by proteomics analysis, or we can sequence all the RNA in it, or we can look for specific proteins. These are markers for extracellular vesicles. We can measure the quantity and size of the vesicles via nano tracking analysis, and we can visualize them with electron microscopy. So we did all these things for extracellular vesicles isolated from blood and from culture media to to really verify that we are dealing with true extracellular vesicles. And I will give you an example of what we use. So we have studied the communication between hepatic stellate cells and Kupffer cells. And I will go briefly to the results. It's a little bit maybe detailed and out of your area of interest, but I just want to show you a little bit how we approach intercellular communication via extracellular vesicles in our laboratory. I already mentioned most of this. This is the activation of stellate cells. So we have the normal liver, and in response to injury, the stellate cells that normally store vitamin A and lipids become activated, and these activated stellate cells produce scar tissue, extracellular matrix, collagen, and alpha smooth muscle actin, and a lot of other factors that promote inflammation. And the question is, all these factors released by stellate cells, are they released as soluble factors, or are they in extracellular vesicles? So we investigated the effect of hepatic stellate cells on inflammation in activation in Kupffer cells. This is an example in which we do cultures of Kupffer cells and stellate cells together. So they are in separate compartments. They are not in physical contact, the stellate cells and the Kupffer cells, but they are able to communicate via soluble factors. So if we co-culture stellate cells with, if we co-culture the Kupffer cells with stellate cells, what you see then is that the Kupffer cells become activated. These are all inflammation markers. These are all cytokines that are increased during inflammation. Interleukin-1, interleukin-6, tumor necrosis factor. And you see that the effect of stellate cells that are in the process of activating on Kupffer cells is to activate them, because the expression of all these cytokines in Kupffer cells, we are measuring cytokine levels in Kupffer cells, is increased when these Kupffer cells are exposed to soluble factors released from stellate cells. So the stellate cells that are in the process of activating activate the Kupffer cells, but we don't know how they do that. So what we did next is that we isolated extracellular vesicles from activating stellate cells, and we added these extracellular vesicles, we added to the Kupffer cells. And again, what you can see is that the extracellular vesicles from activating stellate cells at different stages of activation, day 2, day 7, the main events for activation occur around day 2, and at day 7 they are already activated. But the effect of extracellular vesicles from activated stellate cells on Kupffer cells is again to increase the expression of inflammatory cytokines in the Kupffer cells. So interleukin-6, interleukin-1, inducible nitric oxide synthase, they are all increased by extracellular vesicles from stellate cells. If we remove the extracellular vesicles from the medium that is from the stellate cells, you can see that the effect is still there. So not all the the Kupffer cells are activated by stellate cells via extracellular vesicles, but also by soluble factors outside the extracellular vesicles. Kupffer cells contain receptors on the cell membranes that transmit inflammatory signals into the Kupffer cells to become activated. And the most important receptor that transmits inflammatory signals from the outside to the inside in Kupffer cells is the toll-like receptor 4. There are also blockers available to block this receptor. And what you see in this graph is that if the Kupffer cells are exposed to conditioned medium from the stellate cells, so the soluble factors and the extracellular vesicles, then we see what we expect, the activation of inflammatory markers in the Kupffer cells, interleukin 6, interleukin 1 beta, and INOS. But if we add an inhibitor of the toll-like receptor, the activating effect is completely gone. And this means that the Kupffer cells are activated by components released by stellate cells, released in the extracellular vesicles of stellate cells, in a mechanism that is dependent on toll-like receptor 4, which is the main inflammatory receptor of Kupffer cells. Now, this was an example of how hepatic stellate cells activate Kupffer cells. But as I mentioned at the beginning of this part, the communication is bidirectional. So the next thing we did is to reverse the experiments and study the effect of Kupffer cells on hepatic stellate cells. And markers of stellate cell activation are components of scar tissue, extracellular matrix, and two important components of extracellular matrix and stellate cell activation is collagen type 1, which is a protein present in extracellular matrix and scar tissue, and alpha smooth muscle actin, which is a marker of stellate cell activation. We did the same experiment as in the previous part. We isolated extracellular vesicles from Kupffer cells, normal Kupffer cells, and we added the extracellular vesicles to stellate cells. And what you see is that the stellate cells become activated. The extracellular vesicles from Kupffer cells, if they are added to stellate cells, the activation is increased. Alpha smooth muscle actin goes up, and also collagen type 1 goes up. And also the proliferation, measured here, is increased by the addition of Kupffer cell-derived extracellular vesicles to the stellate cells. Here you can also see that in immunofluorescence. Green is alpha smooth muscle actin inside the cells, and here the green is the extracellular matrix component collagen inside the Kupffer cells. And you can clearly see that there is more green here than here, and also that the green is more intense here than here. Then we took it one step further because we isolated our extracellular vesicles from normal, non-activated Kupffer cells. But we know that in injury, Kupffer cells are activated. So we activated the Kupffer cells in our culture with endotoxin, a bacterial product, lipoprotein saccharide. We added that to the Kupffer cells and then isolated the extracellular vesicles and then added it to the stellate cells. So then we did the same experiment as in the previous cells, but now we use extracellular vesicles from activated and not normal Kupffer cells. And you can see that there is a difference. Again, here in red, we have the staining for collagen, scar tissue marker. And what is important is that the extracellular vesicles from activated Kupffer cells induce more collagen than from the normal Kupffer cells. And the same is true for alpha smooth muscle actin, another marker of stellate cell activation. So this means that activated Kupffer cells are stronger inducers of stellate cell activation and fibrosis than normal Kupffer cells, and that this effect is mediated via extracellular vesicles. So again, it shows you that the state of the cell is important in how they communicate. To use the human analog, here the Kupffer cells, the extracellular vesicles from the Kupffer cells are talking to the stellate cells, and here they are screaming to the stellate cells because they are activated. We also quantified that, so here we see the difference. This is the effect of extracellular vesicles from activated Kupffer cells compared to normal Kupffer cells. We see an increase in alpha smooth muscle actin and also in collagen. So finally, the take-home message is that of this part is that hepatic stellate cells and Kupffer cells interact with each other in a reciprocal state. They induce each other's activation, and once activated, they increase the activation. This crosstalk between cell types is mediated via extracellular vesicles, at least to a large extent, it is mediated by extracellular vesicles. Endotoxin, which is an activator of Kupffer cells present, so a model of liver inflammation, amplifies the effects of the Kupffer cells on the stellate cells, and the activation of stellate cells is mediated to a large extent by toll-like receptor 4. We have done a number of other experiments also with other combinations of cells, for example, Kupffer cells and hepatocytes, stellate cells and sinusoidal endothelial cells, but I think that is too much for today. I would like to show you the people that did all the experiments. Many are from Latin America. We have a good connection with Latin America, so we have Sandra from Mexico, Magnolia from Mexico, Zhongmei is from China, and Mong. They did a lot of the extracellular vesicle work. Joanna from Colombia worked a lot on lipid toxicity in hepatocytes. And I would also like to draw your attention to Camila, who will present next week or in 2 weeks about biomarkers in extracellular vesicles for diagnosing and prognosis of liver diseases. Here she was still a PhD student, but this is a picture of, I think, 4 or 5 years ago. So she's now a medical doctor working in the UMCG, in the transplantation center, and she will be one of the speakers in next week or in 2 weeks. And with that, I would like to end this presentation, and I'll be very happy to answer any questions.
All right. I guess that's the end of the presentation. Am I correct, Professor Han?
Yeah.
All right. First of all, I would like to say thank you a lot to Professor Han for showing us and presenting to us about the importance of liver and how liver disease has taken over the world. Am I correct, Professor Han?
Yes. Yeah.
Yes. And
Yeah,
regarding
I'm
that,
aware that not all of you have a background in biomedical sciences and medicine, so I hope, but that's also a question from me to you, whether you were able to follow this presentation, whether you learned something. Maybe the last part was quite in detail about the experiments that we do in our laboratory, but I hope the introduction to the liver was informative, and I hope it will induce a healthy diet in all of you.
yes, of course, Professor. And maybe for some of us and most of us, we got the big part of what you have given us, to Professor, about the liver, especially how liver disease has taken over all around people in the world. And maybe for the detailed stuff, it's some kind of information that we might have to peel into a little more after the presentation itself. But maybe after this, we're going to have a discussion and Q&A session, if you don't mind, Professor.
Yeah, sure.
Yes, okay. So now for the participants here, we would like to open a discussion and Q&A session for those of you who are interested and would like to discuss what has Professor Han brought to us today. You may raise your hand and I will give you the spotlight.
No question from students?
Maybe, Professor, if I may ask a few questions.
Yeah. So the MC also able have opportunity to ask.
Yes. Maybe, Professor, maybe this is just a light discussion, but whether about the isolation of the cells that you have mentioned. Could you explain more to us about how you isolate those cells and what are the key biometric points that may differentiate those cells that you isolate?
Yeah, that's a very good question. I didn't mention it, I didn't show it, but basically what we do is that we isolate them from rats. The abdomen of the rat is opened, And then we put a needle in the portal vein, which directly goes to the liver. And then we infuse via this needle, we infuse a solution that dissolves the connective tissue inside the liver. So that inside the liver, there is connective tissue that acts as cement for all the cells. So that the cells are basically resting on connective tissue that gives them support and rigidity. We dissolve that. So, and the liver is surrounded by a capsule. So it's basically a bag. And if you dissolve the connective tissue, then you get a bag of cells. So we after this phase, which is performed in the rat, so by flushing a solution through the liver, when the connective tissue is dissolved, we remove the liver, which is then basically a bag of cells. We release the cells by disrupting the capsule of the liver. And then you have a mixture of cells, of liver cells. So the way that we isolate them is in two steps. The hepatocytes are the heaviest cells. So they are isolated by low-speed centrifugation. So then you have a pellet where you have the hepatocytes, and in the supernatant you have the other cell types. And then the other cell types are again centrifuged, but then on a density gradient. Because, for example, the stellate cells, they contain lipids, they have a different density than the Kupffer cells. So after we remove the hepatocytes, we do a density gradient centrifugation, and then we can see the layer of the stellate cells, we can see the layer of the Kupffer cells, and we can see the layer of the endothelial cells. And that is how we isolate the cells and separate them. Yeah.
I see. So it's based on gradients. Am I correct, Professor?
Yeah, yeah.
The gradient,
Mainly on centrifugation and
Ah.
density centrifugation. Yeah, yeah.
That's very interesting. Thank you, Professor.
May I ask something, Prof. Musafir?
Sure.
Yeah. What is let's say because you said that one of the problem is about obesity, yeah, and
Yeah,
for the
yeah.
young generation, especially in Indonesia, we call they like very much to eat gorengan. Gorengan is what is that? Yeah, gorengan. Gorengan is like a fried, yeah, the cakes, but fried with oil. Yeah.
Yeah.
So, is it safe or yeah? Because for me, yeah, we try to reduce the cholesterol. Yeah.
Yeah, yeah. Mm-hmm.
But
yeah.
It's look like it's very very difficult. Yeah, very difficult. Yeah.
Yeah. No. So it's very difficult to give a dietary advice, but in general, you can say that processed foods are unhealthy. So if you have but Indonesia is a country rich in fresh fruits, vegetables, lots of fibers, which is always healthy. If you fry food, so goreng, then usually, or at least in the Netherlands, we use very often saturated fats to fry. And saturated fats are really unhealthy. So if you can replace saturated fats by alternatives, for example, in the Mediterranean diet, olive oil is used very much, and that is very healthy. But I don't know if you in Indonesia have air fryers in which you can fry without oil. So that would be the most healthy then. Yeah. Mm-hmm.
Okay. Okay. But olive oil is very expensive.
yeah, because you don't have a lot of olive. But I think you have alternatives for olive oil in Indonesia as well. I don't know by heart, but I think that as long as you avoid purely saturated fats, so palm oil I think is very unhealthy.
Okay. Yeah. Okay. Nice. Thank you very much.
And in general, for all diets, a little bit of everything. If you only eat hamburgers of McDonald's, it's unhealthy. But every now and then a hamburger is okay. So a varied diet and a modest diet is the most healthy.
Okay, thank you very much.
All right, thank you for the question and discussion from Mr. Tree. And to Professor Han, I have a friend with me here who wants to ask
Yeah.
a question.
Yeah, sure.
Okay. Go ahead, Wayan.
Okay, Professor. I'm here. Want to ask some question.
I don't hear it.
Yes, your voice.
I think you are muted.
No, not mute, but very low.
Okay.
Oh,
Okay.
It's the same as in the other screen. Yeah. Okay. Hello.
So,
Wayan.
Wayan. Okay. Here I want to introduce myself. My name is Wayan Firdaus Putra. I'm here want to know that I'm afraid I missed part of explanation regarding cover cell detection. Could you rationale behind the identification, the feature or marker, biomarker that used to discuss them to another cell in the liver?
So you are referring to bidirectional communication, but I missed a little bit a specific question. Can you repeat that? Was it about this slide?
I want to know how the biomarker that used to identify the Cooper cell from another cell type.
Okay, yeah. So when we isolate cells from the liver, then we have the different cell populations, and then we have to verify the purity of the cells that we isolated. I think that is your question. For hepatocytes, that's very easy. We can distinguish them by morphology because they are much bigger than the other cells. So if you look under the microscope, we can literally count the hepatocytes and the non-hepatocytes just because of their size. And usually the hepatocyte purity in our cultures is more than 95%. For the stellate cells, we use the lipid droplets in the freshly isolated stellate cells to characterize them. So again, what we do is we look at the microscope, we see cells with lipid droplets, which are the freshly isolated stellate cells, and we see cells without droplets, which are the non-stellate cells. The way we isolate stellate cells by density gradient centrifugation, again, we are around 95% purity. Kupffer cells is a little bit different because also they look very much in their morphology to the endothelial cells, and the main contaminant in Kupffer cell cultures are endothelial cells. So for Kupffer cells, we use an immunofluorescence staining with CD63, which is a surface marker on the membrane of Kupffer cells, or CD61, I think. And another marker is CD136. It depends on whether you are looking for human Kupffer cells or rat Kupffer cells or mouse Kupffer cells. But we use immunofluorescence for specific surface markers that are known to be specific for the Kupffer cells. And these are usually cluster of differentiation proteins, CD proteins like CD61 or 63. Now I'm a little bit lost, but and CD136. And then we find out that in the way that we isolate them, the Kupffer cells are also around 90 to 95% pure.
So it can be differentiation by the quantification.
Yeah, of surface markers. Yeah, yeah, that is correct.
Okay, thank you for your answer.
You're welcome. Yeah.
All right. Thank you, Professor. And I have another friend who wants to question, if it's okay
Yeah,
with you,
sure.
Professor. Okay, to Jamie, you may ask your
Yeah.
question.
Can you unmute, Jamie? Yeah.
Okay, Professor. Okay, my question is regarding about your mentioned paper on the earlier presentations about the extracellular vesicle-dependent crosstalk between hepatic stellate cells and Kupffer cells promotes their mutual activations. That's the title of your paper.
Yeah.
Okay, my questions is, since the study found that the LPS doesn't directly activate the hepatic stellate cells, but rather acts indirectly through the Kupffer cells. So, do you believe that the future liver fibrosis therapies should pivot towards the modulating macrophage EV release rather than trying to directly block receptors on the stellate cells themselves?
Yeah, that's a very good question. So I think that if we can modulate the content of the extracellular vesicles in a way that it becomes from pro-inflammatory to non-inflammatory, that would be a great thing. And there is research going on let's say modulating the content of the extracellular vesicles, or even using artificial extracellular vesicles loaded with anti-inflammatory compounds as a kind of therapy. So your question is whether the extracellular vesicles released from Kupffer cells, your question is whether they can be modulated?
Yes.
Yeah. So I think that if you use an anti-inflammatory therapy for liver inflammation, if for example, if you use anti-inflammatory drugs, it will act on the Kupffer cells, and therefore the content of the extracellular vesicles released from Kupffer cells, let's say in treated patients, will be different from untreated patients. I expect that medication, so a therapy, will affect the content of the extracellular vesicles released from the target cell of the drug. Yeah, I think that is a very good question to investigate whether, let's say, in patients on anti-inflammatory therapy or no therapy, whether the content of the vesicles released from the inflammatory cells, in the case of the liver, the Kupffer cells, is then different. And if yes, then we can also identify the compounds that activate the stellate cells. Yeah. That's a nice question. Yeah.
Yeah. Okay.
You can come to
Thank you
Groningen
for
to investigate this.
yeah.
All right. Thank you, Jamie, and thank you, Professor, for the answer. Maybe if one or two more participants have a question, you may raise your hand in the Zoom meeting, and we will give you a chance to ask your questions to Professor Han.
No question? If not,
Okay.
then we can, yeah.
Maybe I can say a few words as conclusion. So the next speakers will talk also mainly about extracellular vesicles. Extracellular vesicles as a tool, as novel biomarkers for predicting the outcome of liver diseases. Also, extracellular vesicles to modify in a way that they can be used for therapeutic applications. And also more about drug targeting. I know that your background is not exactly in biomedical sciences, but I would also like to mention that within our university, and Professor Tree knows that very well, we have a large department of biomedical engineering and other engineering, other applications of engineering for medicine as well. My talk is a little bit outside your scope, I guess. But we do a lot of research in addition to this in terms of biomedical engineering. For example, we also work with prosthesis, implant technologies, biomaterials, 3D printing, etc. So that I would like to mention at the end. Yeah.
All right. Maybe since Professor Han has concluded our session for today, thank you for the conclusions, and thank you most of all to Professor Han for giving us the presentation and giving us knowledge about liver disease and liver structurals and all of that. And maybe even if we don't 100% get it, but it's also useful information that we have to learn that in the upcoming sessions, we might get a lot of more useful informations about it. And before we close our session for today, I would like to invite everyone here in the Zoom meeting to open your camera for us to take a documentation, if you don't mind, Professor Han.
Yeah, sure, no problem.
All right. To the committee, you may start taking pictures. All right, the committee will start taking pictures. So now for slide 1 in the Zoom meeting, we'll take a picture in 3, 2, 1. Okay, now we'll move on to slide 2. 3, 2, 1. Okay. Moving on to Zoom 3. 3, 2, 1. Okay, I think that's all for our agenda for today. And one more time, I would like to say big thank you to Professor Han for coming to this Zoom meeting and giving
it was a pleasure to do this. Yeah.
Yes, it was a big pleasure for us as well. And I would like to say the biggest thanks from Biomedical Engineering Department in TS because of all the useful informations, all of the knowledge that we have gained and the knowledge we will gain in the upcoming guest lecture series. Maybe Mr. Tree would like to add some statements.
No, we have many discussions with Prof. Han Mosagi. This is for special for you, for the student. So, thank you, Prof. Mosagi.
Okay, thank you very much. Thank you very much for giving me the opportunity to present this lecture. And good luck with you all. And hopefully I will see you or in Surabaya or in Groningen. So
yes.
Yeah.
We'll hope to see you as well, Professor.
Okay.
Yeah. Okay.
Thank you.
Bye-bye.
Bye-bye.
All right, everyone. Thank
Bye-bye.
you. Thank you, Professor Han. We'll end
Yeah.
the meeting. Thank you, everyone.
Good.